So this morning is on type 3 secretion. Again, just to show you where this sits, so this is here, third of my 5 lectures, goes together with the one on the bacterioflagellum. We've got 2 more from me coming up later this week, early next week. So at the end of this lecture, you should be able to describe the structure, function, genetics, biosynthesis of type 3 secretion systems, should say including regulation of type 3 secretion, protein secretion and gene expression, and provide examples of diversity and wider importance. So if we look at these images I put up here, you might think there's nothing really that connects them. We have here the spread of the Black Death through Europe back in the Middle Ages, bubonic plague. Here the need for new chlamydia screening in Ireland. This was a government minister who lost her job for talking about the risk of salmonella in eggs, children dying from E. coli and dysentery. In fact, all of these different kinds of infections are linked by a common mechanism of pathogenesis known as the type 3 secretion system, and that's what I'm going to talk about today. So just to return to this rather busy image from Michael Desvaux's review, we have, when we look at gram-negative protein secretion, a division into those secretion systems where you have 2 steps, one crossing the inner membrane into the periplasm and then going out through the outer membrane, and those where there is a single step in which the secretion system spans the entire cell envelope in one go. And here is type 3 secretion here as an example of such a system where there is no periplasmic intermediate. Proteins go all the way out through the cell and in fact go beyond that into other cells, all in one step. Now, we mentioned flagellar type 3 secretion in the previous lecture. In kind of nomenclature terms, there's a bit of prevarication, confusion in the literature as to whether you should use the term type 3 secretion for flagella, the flagella protein export apparatus. I think you should. And we should talk about all the others as non-flagellar type 3 secretion systems. These non-flagellar type 3 secretion systems as target of today's lecture are widely distributed among gram-negative bacteria. We don't ever see them in gram-positives, unlike the flagella systems. And they're responsible for the translocation of what we call bacterial effector proteins into eukaryotic cells. So they have a very particular function. And those effectors, when they get inside the eukaryotic cells, target conserved parts of the eukaryotic cell biology. And we know that there are many examples of effectors that will act on humans, on plants, on insect cells that are homologous to each other and are obviously targeting parts of the conserved molecular toolkit of all eukaryotes. Now again, this is a very crowded slide, but it's just to give you a flavour of how widespread type 3 secretion is and what a flexible tool it is, deployed in lots of different settings. That first came to prominence, obviously, as a cause, as a contribution to human disease with human pathogens, some listed here. But more widely now, we recognise it in fish pathogens, insect pathogens, in various symbionts. Sedalis is a symbiont of the tsetse fly, of great interest because of the role that plays as a vector in sleeping sickness. Various plant pathogens. In fact, much of the fertility of the soils across the world depends on type 3 secretion because leguminous plants rely on an interaction with bacteria, which in turn relies on type 3 secretion. We've got pathogens of fungi. And as we sequence more and more genomes, and we're working our way through the whole biosphere in terms of sequencing everything out there, we actually keep discovering new type 3 secretion systems in genomes where we don't actually know what the function is. We don't know what the target is. And it was a bit of a mystery there. Now, you've come across the flagellar basal body and the hook before. The virulence-associated type 3 secretion system, or non-flagellar type 3 secretion system, has very similar structures. There's clear homology at the level of the protein sequences, in the level of the structures of many of the proteins, and in the way in which they are deployed together in this multi-protein complex. So we have this structure here spanning the inner membrane, spanning the periplasm, then we have a ring here spanning the outer membrane. This is where there is some deviation. The actual proteins involved in spanning the outer membrane are distinctively different between flagellar and non-flagellar systems. Then you have a filamentous organelle coming outwards. It's called the needle in this case, capped with various proteins. And then you have a ring which is inserted into the host membrane. We'll say more about that later. And again, the structural biologists with their cryo-EM and other reconstructions have delivered these very beautiful renditions of the whole system. Here's another figure taken from a review by Cornelis, just highlighting those similarities and differences between the flagellum and what he calls the injectosome. I'm not a particular fan of that phrase. It sounds as a bit of a neologism too far for my liking, but you will see this sometimes used, this term injectosome used as a description of this complex here that's produced by the type 3 secretion system. So in the simple form you have this so-called needle complex. You have the basal, the equivalent of the basal body, then you have a needle coming out from that, extending out from the cell. That needle can be capped by longer filaments, and we'll say more about that in a moment, or in some cases in plant pathogens there seems to be an especially elongated needle which is much longer than we see elsewhere. Now, what does type 3 secretion do? Well, there are effectively 5 components and 5 functions. We have regulators, chaperones, the secretion apparatus itself, translocators and effectors, and there are 5 functions. Basically, these proteins, the system exports proteins across the bacterial cell envelope, brings the host cell and the bacterial cell close together. It translocates proteins all the way through this continuous space, if you like, between the bacterial cell cytosol and that of the target cell. And those proteins then go on and subvert host cell functions. Now, this was supposed to work as a, here we are. Yeah, so you have here chaperoning of the effectors to the apparatus, delivery to the apparatus. They go through the apparatus in unfolded form, and then when they reach the target cell, they fold up, and then they have their effects on the target cell. Now, one of the problems that we have in this field is that there is no stable, agreed nomenclature for the components of these systems, even when it's apparent that the various components are all homologous to each other. They have equivalencies in different systems. They have different names in different systems. So here are 4 of the most well-characterized systems, and you can just see the confusion that we have here in the literature. So I guess the clearest thing is that in E. coli, they have more or less followed the Yersinia description. So a needle subunit that's called ISC-F or YSCF in Yersinia is called ESC-F in E. coli. But the equivalent thing in the Shigella system is called MIXI-H, and in Salmonella it's called PRUG-I, and so on through the list. So this is just an annoyance. People have tried to standardize things, but you get people who work on Shigella, they stick with their Shigella names. The people who work on Yersinia want to stick with their names and so on. And this has never really been sorted out. And it does just make it hard to interpret the literature. Now, the structural biologists, as I say, have been very busy with all the structures of the various components. We're now getting, we're not quite at the level as people are with the bacterial flagellum in having a kind of atomic resolution model of the whole filament that goes on for microns in length. But we are getting to see how various things fit together, and these various beautiful structures kind of seen here in this figure, accounting for the various macromolecular structures there. So, for example, we have this model of this ring here, this inner membrane ring, the SJ ring, where we can see quite how those various components slot together. When we look at the genetics of these type 3 secretion systems, we generally find the genes in the same kind of order, clustered together in each individual secretion gene cluster. This is my favourite system, the LEE, the Locus for Enterocyte Effacement cluster here. And here it is compared with the one from Yersinia, the YopN-TyeA cluster. And you can see that there are some runs of genes which are in common. Occasionally things get flipped round, operons move about, but there is some kind of, you can see there's some kind of method to the madness in terms of the way evolution has worked. There are some counterexamples. In chlamydia, we see the genes for type 3 secretion scattered much more around the chromosome, not clustered in quite this obvious way. As I will explain a bit later in the talk, we have discovered now that you can get the effectors of type 3 secretion encoded elsewhere, not in the same locus as the secretion system itself. Now, regulation of type 3 secretion gene expression is very tightly regulated, as you might expect. I mentioned that there are all these equivalencies, homologies between the secretion systems and different systems with different components. So you have, in one sense, you have this conservation, but on the other hand, you have intense variation in the regulators. This is the most variable part of the type 3 secretion system. So although the building bricks are the same in different systems, the ways in which they're regulated are actually very different from one system to another. And this table here just gives you a highlight of the outline of some of the various factors that you can get, transcriptional regulators you can get in different secretion systems. And this is by no means comprehensive, but it just shows that there's a whole range of different regulators, and there is no one-to-one equivalence of what's going on in the Shigella system to what's going on in the Salmonella system to what's going on in the E. coli system, and so forth. There does appear, however, to be one theme that seems to be conserved, which is that, as with the Flagella biosynthesis apparatus, there is, to some degree, or varies from one system to another, there is some coupling of gene expression to protein secretion. So the system, in some ways, recognizing where it's at in terms of the program of biosynthesis of the system, and is regulating gene expression and regulating also protein secretion in time with that. So again, this kind of just-in-time mentality does actually permeate these systems as well. Now, looking at the various proteins that contribute to the mechanism of type 3 secretion, let's start with the type 3 secretion chaperones. So things that are going to go out through the system, they need to be protected in the cytoplasm. They also need to be guided, delivered to the secretion apparatus. And for this purpose, we have these type 3 secretion chaperones. Now, some people complain we shouldn't really use the term chaperone because these are not like Growiel and so on. They don't require ATP or anything like that. They're not enzymes. They're just basically binding to the proteins, coating surfaces that will prevent them from folding up properly, folding up completely in the cytoplasm before they're ready. They have to go through the system in an unfolded way. So they're preventing rapid. If you take the chaperones out, you either get rapid degradation of these of their partners, or aggregation, or you get reduced secretion of the secretion substrate. And talking in very general terms, there are two main structural or functional classes of these chaperones. Class 1, we could say, are the chaperones of the effectors, the proteins that are going to go all the way through the system and into eukaryotic cells. Class 2 are chaperones of the translocators. The proteins are actually involved in getting proteins across the boundary between the bacterium and the host cell and through the host cell plasma membrane. So when we look at the class 1 chaperones, we have this complex alpha-beta fold, which the fold is conserved between different chaperones in this class. So the sequence similarities actually can be quite hard to identify in some of these, but we do at least have some models now of how these chaperones are binding to the effectors, binding as dimers to one component of the effector, typically towards the N-terminus of the effector. There's a chaperone binding domain there. Class 2 chaperones, a few years ago with Klaus Futterer here and myself, we actually came up with a homology model which turned out to be entirely true, showing that these actually have a particular fold known as a tetratrichopeptide repeat. This is an array of alpha helices in a kind of concertina arrangement, and we were able to show, when we mapped the residues involved in interactions with different substrates, that different faces of this fold were being used to interact with different substrates separately. This has since been confirmed with structural studies, experimental studies. Now, as I said before, these secretion systems act as molecular syringes, and we have this basal complex with an ATPase, just like in the flagella system. We have the needle. In the E. coli system, we have a special extension called the ESPA pilus, which more later may be present in some other systems. We find homologs, but we've never seen it deployed as a pilus. And then a translocation pore. So these are grabbed out of various journals. Here's a model of the needle complex from Shigella. I think this is from Susan Lee's group. And then here is an atomic force microscopy image of the pore that is formed in the eukaryotic cell plasma membrane. Biosynthesis and secretion of this whole apparatus is a bit like the flagellum. You've got, the whole thing is hollow, and you have things, the various subunits going in from one end, going all the way through, and then being assembled at the distal end. And the thing is growing from the distal end all the time. Unlike the bacterial flagellum, it's not spinning at the same time. Now, one of the key concepts that you need to get a handle on here is that when these effectors are secreted through this system, they're not just chucked out into the external medium into in soluble format into the supernatant of in a culture in the lab or into the environment outside. They undergo what we call polarized secretion, which means that they are not released to the external milieu, they're not accessible to proteases, that kind of stuff. They actually pass through this conduit that goes all the way from the bacterium into the host cell. Now, this, I'm just not going to go into detail here, but I mentioned earlier this kind of just-in-time mentality, that there is this program of events where you get switching going on as the system is being produced. And there are at least two switches that have been identified, where we have these early substrates, middle substrates, and late substrates. And there's a switch in the specificity of the secretion system for particular proteins to go through it that takes place between early and the middle substrates and between the middle and the late substrates. Again, I could go into enormous detail about this, but I suspect that would not be appropriate here. But we have in that first switch a Fly-K homologue. If you remember this molecular tape measure we mentioned in the flagella system, it's clear that there is an equivalent. There's actually a homologue in these systems as well, and that interacts with the apparatus, measures, we assume, again, measures the length of the apparatus and then changes specificity. There's also another protein called YSCU, which is part of the basal apparatus on the cytoplasmic side, that gets cleaved at a particular loop with this amino acid motif NPTH in it, and that is also part of this switch at that stage. The second switch is controlled by a protein pair in Yersinia called YopN-TyeA, although I showed a few years ago that these actually have homologs in almost all these systems that act as a single protein. Yersinia is a bit of an unusual one in having them broken into two. And what this does is it's a bit like an effector that is trying to get secreted and blocks up the system. And then only when there is this contact made with the host cell, and there's some kind of, it looks like there's a signal, a low calcium signal that passes through the system. Once you've got this contiguous conduit here, that particular gatekeeper protein is released, and then all the other effectors can go through as well. So the effectors, when they get through, what do? Well, there are probably at least half a dozen lectures that could be given on how these type 3 secretion effectors have their effects on cells. But as time is short, I'm just showing you, here's lots of interesting structural biology gone on solving the structure of these effectors. And in general terms, we can identify a variety of targets of subversion for these effectors. So these effectors they're not all out toxins just trying to kill a cell. They're trying to actually manipulate the cell to the bacterium's advantage. And they manipulate things like the cytoskeleton, particularly actin pathways, inflammatory responses. In epithelia, they can influence tight junction barrier function, cell cycle, mitochondrion, can influence apoptosis. And as I mentioned before, they're often targeting the most conserved parts of the eukaryotic cell toolkit. Okay, so far we've spoken in very broad terms about type 3 secretion. Let me just now focus in our efforts a bit more and talk about type 3 secretion in E. coli in particular. We mentioned salmonella in passing. But in E. coli, we have this locus for enterocyte effacement, or the LE, which is found in enteropathogenic E. coli and enterohemorrhagic E. coli. There is also, as we described a few years ago, there's this defunct system, which is named ETG2 for E. coli type 3 secretion system 2, which occurs in a wide range of E. colis as a gene cluster in various stages of disrepair. And so far we've never actually found that an intact gene cluster produces a functional system, but by homology it's clear that there was once a functional system. Shigella, which we said in the E. coli lecture, is a kind of E. coli. This has its own particular type 3 secretion system encoded on a virulence plasmid. That system is often known as the MIXI-SPA type 3 secretion system. And then we have the flagella systems of E. coli as well, which are in effect type 3 secretion systems. So you can see there's a wide range of these different secretion systems in E. coli. In Salmonella, we also have these two systems, the so-called SPY1 and SPY2 systems, named after the locus in which they're encoded, the Salmonella pathogenicity islands 1 and 2. Now, I'd like to be able to give you lectures on those, but in the interests of time, we're going to have to pass over that. In previous years, we tried to ram that in, but I think we overdid it. So I'm now going to focus in on the LE. So if we look at the LE, and we look at the whole of the chromosome of E. coli 0157, and we colour in the chromosome according to the GC content of the chromosome. So if you remember that the GC content can vary independently of, so although the amount of G and the amount of C is going to be the same in any chromosome, the ratio of GC to AT is free to vary. So if you think about the genetic code and that last residue typically being the site of redundancy, like something like glycine can be GG anything, it can be GGA, GGT, GGG, or GGC. What you find in bacteria is that they typically have, for the whole chromosome, a kind of average codon usage, average GC content. And if you do these kind of maps, where you look at a little sliding window around the chromosome, you can often see interesting regions of deviating from the common average background. And you can see it here. There's a little wedge of lime green here, and that's because the Li actually has this very low GC content compared to the rest of the chromosome. That is taken as a hallmark of the fact that it's horizontally acquired recently, has not been able to adapt to the background GC content. And focusing in more detail, if we say compare EHEC or EPEC with an E. coli that doesn't have the Li encoded, the Li in it, say K12, what you see is something like this. That in red here, you've got things that have orthologs. We might call those the backbone of the species. Those are present in all of the members of the species. And then we run into this area where there are no homologs elsewhere. This is showing the same thing in a different depiction here. This is a K12 region. This is in EHEC or EPEC. You see the Li appears as a wedge, if you like, that's been shoved into the genome there. And this is just showing the GC content focused in much more detail now. And again, you can see pretty much every gene in the Li is showing this GC deviation, extreme low GC content. So this type 3 secretion gene cluster, what's it doing? We've mentioned it before, the locus for enterocyte effacement, or the Li, mediates this attaching and effacing phenotype, which you can see in these electron micrographs here. So you have the EPEC or EHEC here in very close proximity to the eukaryotic cell. You get the induction of influence on actin, cytoskeleton, the structure of this pedestal. The intimate attachment is the result of this protein called intermin, and as I'll explain in a moment, that is interacting with another protein called TYA. So we have attachment to the host epithelial cells, we have subversion of the cytoskeleton, we have this effacement, a bit of a fancy word for describing destruction of the brush border microvilli and this pedestal formation. Now, as I've mentioned in passing, we have in this system a kind of variation on the common theme, in that we have all this basal complex here, we have all the same components that you get elsewhere, the ATPase and so forth, these rings spanning the membranes and so forth. But in addition, there's this long filamentous organelle between the needle and the translocation pore, which is known as the ESPA pilus. And this is a bit like the flagella filament, in that it is the result of polymerization of a single protein called ESPA in this system, which is added to the filament at the far end, just like in the flagellum. In fact, a friend of mine, Gadi Frankel, was the first to show this he took ESPA from EPEC and ESPA from EHEC, and he expressed them, he made one of them under an inducible promoter that he controlled, and he managed to switch one on halfway through the process, and he could see, using an antibody, the switch in the synthesis, showing that actually new things were being added at the far end. Here are some pretty pictures of electron micrographs of the ESPA pilus associated with the actual basal body. You can see them like little rivets that have been ripped. These have been ripped out of their normal context in the cell envelope, and you can see here the arrays, various lengths, going up to getting on for, well, in fact, going up for a micron long, 700 nanometers in length. This is from Stuart Nutton, and Rob Shaw, I think, was involved in this, looking at these filaments. So these filaments were stained with an antibody raised to purified ESPA protein. And you can see that what you do here is you have an antibody with a little gold particle on the end of it, a couple of gold particles. So this is immunogold stain, and you can see here that you get the filament here, the ESPA filament, nicely decorated with these all the way along. And then, in fact, there's just at the bottom here, there's this kind of scraggly neck. That is the actual needle that is seen in other systems. It's just this little bit of needle before you get to the ESPA pillars. And you can see these ESPA filaments actually contacting the host cell, bringing the bacterium into association with it. And in fact, you can, in this electron micrograph, you can see them effectively tunneling. This underneath here is the plasma membrane of the cell to which these bacteria are attached, and you can see that ESPA filament diving into it. And this is clear evidence that it's mediating this conduit into the host cell. And here's again, you can see this is in a transmission here, and you can see this here decorated with the anti-ESPA antibodies contacting a host cell. Here's a pretty picture from Rob Shaw, who used to work in my group. These are some EPEC with their ESPA pili, and they're in contact here with red blood cells. This is totally non-physiological. They don't meet red blood cells normally, but he has shown that actually you can use the red blood cells as a model for enterocytes. You don't actually have to have enterocytes. You can use these, and they will go on and deploy the ESPA filament and interact in exactly the same way. He's obviously gone and coloured that in as false colour. It's not real, it's an electron micrograph. Now, just in the last few minutes of the talk, I'm just going to take you into some of the research that we did a few years ago. Before that, I'll just give you a summary video, actually. I just remember we've got a video to come. So if we look at the simple model that we had maybe 7 or 8 years ago, we had this idea that here was the lead. It encoded all of the proteins needed to make the secretion apparatus, to make the translocation apparatus, and so on. And it also encoded 7 effectors. And it was thought, well, that's it, that's what it does. Those 7 effectors, it's a self-contained system. And there's actually, I guess, an assumption that kind of has grown up over the years with the definition of pathogenicity islands, this idea that a pathogenicity island comes in and gives a bacterium a quantum leap up to a new virulence phenotype. And the assumption was that the Lee would come in and do that to E. coli. And it would, kind of implicit in that assumption was it was self-contained. It was just doing its own thing, had everything it needed to do that job. Now, one of the interesting things that was discovered in the mid-90s, so we have this intimin that was known to be on the cell surface, the outer membrane of the E. coli, somehow mediating intimate attachment. But what was it interacting with? And for many years, people thought, well, it must be interacting with a host protein. And we just need to find what that host protein is. And then there was this surprising finding that it wasn't quite that simple. What was actually happening was that the bacterium was producing a protein called TYA, which was being translocated through the secretion apparatus and then integrating itself into the plasma membrane of the host cell and then acting as a receptor for intimin. So if you like, the bacterial cell was controlling both sides of the interaction. So this protein TYA was translocated into host cells, inserted into the plasma membrane. It's got this hairpin structure with these 2 transmembrane domains, an extracellular loop that's sticking outwards, and then inside the cell are the N and C termini pointing downwards and interacting with stuff in the cytoplasm. So the extracellular domain is what interacts with the outer membrane protein intimin on the bacterial surface. And in fact, what happens is that intamin and TYA dimerize, but they're interacting with different partners. So in a way, they kind of stitch the cells together, or zip them together, if you like. They form this kind of reticulate array. So here's some figures from journal articles by Jim Caper and others. The model was that you have the epithelial, you have EPEC here, an epithelial cell. It's got ESPA, it's got these bundle-forming pili as well, and intamin. Intamin, I didn't say, is actually encoded in the Li, along with, so it's not actually part of the secretion system or a target of the secretion system, but it's part of this whole get-up of how the cells come together. You get the deployment of the ESPA filament contacting the cells, releasing TYA, that having various effects on actin. You get the intimate attachment where intamin and TYA come together, and then you get the rising up of the pedestal. This here is a thing showing the interaction between the various domains that have now been solved of intamin and TYA, and this doublet interaction there. TYA, though, is a very versatile protein because in addition to acting as a receptor for intamin on the outside, those loops outside the cell, the stuff inside the cell, those two domains inside the cell, are orchestrating all sorts of cytoskeletal arrangements. And in fact, TYA is the thing that is largely orchestrating the formation of the actin-rich pedestals underneath the bacteria. And in the early stages, there was a lot of research on this. There was a lot of interest in the ways in which phosphorylation was going on with TYA, and then interactions to various parts of the actin cytoskeletal network. So in one kind of TYA from EPEC used this NCK as a linker. In EHEC, there was another effector that acted as a linker to NWASP. But somehow or other, TYA was actually interacting with the actin cytoskeleton. So now I'm going to show you a movie from Brett Finlay's group from a few years ago, which summarized all that, the state of our information around the turn of the millennium, if you like, as to how the type 3 secretion system, intamin and TYA come together. So let me see if I can plug that into the sound. Can I do that? I should be able to. So you get the sound as well. No, I'll tell you what I'll do. I'll just put the microphone close to my speakers. That'll probably work.
Here's our bacterium here, and behind it will be these long strands that are the bundle-forming pillars. They're going to adhere to the microvilli, these wiener-like structures. This is what your normal intestinal cell surface comes along here. Bacterium then causes these things to disappear. It then starts to nestle down on the surface. It's locked in. And then it's going to come down, and it's going to set up camp right there. We know that happens because we can see this in electron micrographs, and the microvilli all disappear, and boom. Okay, so we're now stuck in the intestine. Bacteria are here. We're going to zoom in. Here's our type 3 secretion system, right? This thing will poke out. We know a protein comes out. We know this protein. And then it inserts two more proteins that come out and down this tube, and they go into insert into the host membrane. So these green proteins will sort of form like a donut pore, and this is kind of the tip of the syringe. Then we have to fire in the receptor, the TYA molecule, and you'll see it as this red molecule coming down the chutes. And it gets then fired into the host cell. Then it goes into the host cell. We don't know if it goes through the cytoplasm, goes straight in the membrane. We have to figure this out. You'll see a little blue dot. That's a tyrosine phosphorylation event. TYA gets modified. Then it ends up in the host membrane. So here we're piling the receptor into the host cell. And then we then have to dock TYA with intamin. So we think this tube-like protein retracts. And what will that then do is bring the bacteria down into contact. Here's intamin up here, blue TYA here. Houston, we have contact. We have now landed. Bacteria is stuck on this cell. And now we're going to go inside the cell, and we're going to start to see the face of TYA that's facing. It recruits cytoskeletal proteins and these little donut-shaped things we we're starting to understand more. Then you're going to see little yellow beads come flying in. These are actin monomers. They then get together, and it looks like a string of pearls. They polymerize. See these things flying in, and then start to get longer and longer. Actin does this for a living. It forms these long projections, strings of beads. And what that will start to do is start to put a force on this thing, and you will then start to see the bacteria actually raise up on the cell. And it's because there's actin pushing underneath it that it actually pushes it up into the cell. There it goes. And then it starts to push up. Bacteria starts to raise up. It's now building its throne. I'm going to sit up on top of this thing and rule out over it. If this doesn't want to make me be a microbiologist, I don't know what will. Isn't this a thing of beauty? And then it just continues to rise and rise and rise. And the end result is we then end up with the bacteria sitting up on this throne, shall we say, to rule out over the mammalian cell. And that's what we see in disease.
Okay. So that was our understanding of things in the early 2000s. I put this up just so it will go into the handout for you, so that when you come to revise this, you'll be able to look at this. I'm not going to talk your way through that, but that's basically a transcript of what the guy was saying, Brett Finlay was saying more or less. 2006, I started looking at this issue, and we thought, well, are there actually effectors outside of the Li? And if so, how many of them are there that are encoded outside the Li? Now, it's clear that there were some. In 2006, a handful have been discovered. They're listed here. I'm not going to go through each of them in detail. In fact, Brett Finlay, who you heard speaking a moment ago, actually pioneered this area. He came up with a related organism, Citrobacter rodentium, which has a Li locus in it. Doesn't cause quite the same disease, but he discovered that there were some so-called non-Li encoded effectors that he found in the chromosome of that particular pathogen. They called them NLEs, so NLE-A and so forth. He actually, what he did was he knocked out every gene in the Li, in that mouse pathogen, and he found, so I mentioned YopN-TyeA earlier. What he found was that there was the equivalent of this thing called Sepl in the Li, which was acting as the gatekeeper. And if you knocked out that gatekeeper, the bacteria, which normally would not secrete effectors into the supernate, normally they just secrete the translocators, ESPB and ESPD, into the supernatant. But if you knock that out, then they suddenly start secreting effectors very effectively. And what he did was he just characterized the new proteins he saw being secreted, and he described them, just gave them names, didn't really do any further work on them, just called them NLE-A through to NLE-H. Now, at that time, it was clear that in a related, in a system with a related type 3 secretion system, an organism with a related type 3 secretion system, Pseudomonas syringes, a plant pathogen, it was clear that there was a large number of effectors encoded in the genome, over 70 or more. And it struck me at the time that this was a bit odd. You know, why have you got 70 effectors in one organism and only 7 or 6 or 7 in the Li? And even if you allowed a few of these extra ones that have been described, it still looked a bit odd. Now, you can argue, well, those kind of intuitions, you shouldn't think that way inside something. Maybe the world sometimes is just the way it is. But it got us thinking that, well, maybe there were more things in EHEC or EPEC than we expected. And so we did a piece of research where we collected up all the known effectors that had been described. I think there were about 300 of them. And we then searched using various search tools, BLAST-P and SciBLAST, against the entire EHEC genome. We then looked at the context of everything we found, and we looked at genome comparisons. And we also brought in some experimental approaches. We did a little bit of that here. A lot more was done in Gadi Frankel's lab and in Tobei's lab in Japan, Toru Tobei's lab. And what we found when we put all this together was actually there were a large number of new effectors out there. So with the homology searches, we found 62 candidate effectors. When you look carefully at these, when you look at the homologies and so forth, we could see that in some of them there were frame shifts. or premature termination. So he said, well, those are not likely to actually encode a functional protein, so those are pseudogenes. But we found 49 potentially functional effectors, and these were clustered into what we called exchangeable effector loci, or EELs. I was very pleased to come up with that acronym because it's the opposite of Lee backwards. Proteomics, the guys in Japan there made a Sepl mutant similar to the way that Brett Finlay did, and they just took slices and they cut slices and they mass spec those and identified 31 secreted effectors. And then all three groups did these so-called translocation assays, where you take a gene that you suspect to be a translocator and you fuse it to some kind of reporter, and then you detect the movement of that reporter into a eukaryotic cell. And I won't go into the details of how that's done, but basically that confirmed that 29 of these effectors were indeed translocated. So we ended up with 62 candidates, 49 potentially functional, and 39 of them actually experimentally confirmed. And we were very surprised, because actually there's over 20 different families of effectors in E. coli 015787. We found homologs of things from Shigella, from the virulence plasmid in Shigella, which are poorly characterized. We found effectors from plants with homologs in E. coli, homologs of Salmonella effectors, and we looked at these NLEs, which have been described by Brett Finlay, and we found multiple copies of some of those. Surprisingly, there was one particular protein, NLEG, which there were 14 copies of the gene encoding that protein, or members of that protein family in the genome, which is a new finding, quite unexpected. As I say, we have these EELs, or exchangeable effector loci, a couple of dozen of them scattered through the chromosome, and they fell into two main categories. One group were not related to phage at all, not obviously related to phage, and they appeared to be lineage-specific single gene insertions. And we often saw these as pseudogenes, and we don't know, but we think that they may have been targets of the old ETT2 system, which is now defunct. And maybe they're just baggage of history, they don't actually have a function. But more interestingly, we found these phage-encoded EELs. So these are clustered effectors of effector genes carried as passengers within prophage. So we know that lambdoid prophages are very common in E. coli generally, and in pathogenic E. coli. And there are 13 of them in E. coli 0157. And 9 of the 13 were actually found, when we look carefully, to encode type 3 secretion effectors. And in those prophages, all but 3 of what we call passenger genes were actually effectors when we look carefully. Put it another way, when we look at all these effectors, the majority of them are phage encoded. So this here shows you the chromosome of EHEC again, with the Lee coloured in there. And then just one side of the chromosome, because it won't fit on the slide, I've just started to mark in some of these EELs as we go around. And you can see that they're scattered through the chromosome. We've got this phage 3, phage 4, phage 6, phage 8, phage 9, phage 10, all of these encoding effectors. And the same pattern goes all the way round as well, scattered across the chromosome. And looking more carefully in detail here, if we actually focus in on the ends of these phages, what you see is this very interesting pattern in the GC content. So these kind of reds and oranges here have slightly higher GC content than the bacterial chromosome, the E. coli chromosome. And then, adjacent, you have these green ones, which suddenly have a very, very much lower GC content than the average for the chromosome. And the arrows, blocked arrows there that are shown as large arrows, thicker arrows, each of those is an effector as judged by homology. So you can see there's this remarkable congruence here of evidence from genomic context, GC content, and homology, all fingering these particular effectors at the end of these prophages. So there's like a passenger compartment in the phage genome, if you like. So the conclusion of this work is that the Lee is not just connected to other loci in the host chromosome, But because those loci are actually prophages, that actually some of which could be active as phage, there's this vast phage metagenome out there. And if we compare different strains that carry the Li with each other, we see there is actually considerable variability in the deployment of different prophages in the genome. So there's obviously lots of phages coming in, going out, dropping in, going out, and so forth. And in fact, Scott Beatson, who was working for me at the time, came up with this little quip here, which is that, given that the Li itself is probably phage-derived, it's not an integrase in certain situations, the Shigella genes are phage-derived, maybe we should see the infections caused by E. coli as not as infections of E. coli, but actually infections of phage, and that E. coli is just the mediator of a phage-human interaction in terms of pathogenesis. We wrote all this up in a paper that appeared in PNAS in 2006, and we drank some champagne to celebrate the fact that we got that paper out. So, I've finished now. In summary, we have these type 3 secretion systems, multi-protein complexes connecting bacteria to host cells. They mediate protein secretion and translocation from the bacterial cytoplasm to host cell interior. The effector proteins subvert cellular functions. An interesting point is that these effectors are actually of interest to a wider field, because people interested in the actin cytoskeleton can actually use these effectors to sort of interrogate the systems they work with. And here's a quote from Stan Falco, one of the godfathers, which basically points out that bacteria have been doing cell biology for a lot longer than humans have. They've been studying eukaryotic cells and how to subvert them for billions of years rather than the mere 100 that we have. Right, that's me finished for today. I'll take any questions if there are any.